So I'm Steve Hill. I'm Professor of Molecular Pharmacology in the School of Life Sciences at the University of Nottingham. And I'm also Director of the new Centre of Membrane Proteins and Receptors, which is a new and exciting collaboration between the University of Nottingham and the University of Birmingham in the Midlands. So my interests are in a cell surface protein, a G protein coupled receptor, which recognises and responds to neurotransmitters and conveys signalling to the intracellular environment of the cell through an interaction with the G protein, which hence its name. And we're increasingly interested in how we can target those proteins with new drugs for particular therapeutic implications, and increasingly how they are affected by being in larger complexes and how they vary from one cell type to another, which may give us new opportunities for targeting with novel drugs. So NanoBRET and the introduction of the bioluminescent protein NanoLuc by Promega has really revolutionised what we do in studying the direct binding of ligands to receptors. It provides a very bright luminescent protein that we can attach genetically to the N-terminus, or the outside part of our target of interest, the G protein coupled receptor, and allow us to monitor close proximity of drugs as they bind to that protein by transferring energy from the luminescent NanoLuc to the fluorescent protein, which is what NanoBRET is. And the real big advantage over radioligand binding techniques for us is that we can apply that to living cells, and we can also study it in real time, so we can monitor kinetics of the interaction for the first time, really. So the collaboration with Promega has been pretty essential for us. They provided the NanoLuc luminescent protein, which they developed from a protein generated by a deep sea shrimp. And they were able to engineer it to be a small protein, which allowed us to attach it to our targets of interest. And later on, I guess that the original approach came from Promega to us because of our existing fluorescent ligand technology. They asked whether we would be interested in collaborating to see whether we could use the technology to look at ligand binding in real cells. And we were able to show that even with our very far red-shifted ligands, we got such a fantastic signal to noise that it's really begun to overtake radioligand binding experiments in my lab. And then with a second collaboration, we developed approaches to label larger proteins with some other unique technology that Promega had. We were able to label VEGF and show that the NanoBRET approach worked just as well with receptor tyrosine kinases as it did with GPCR. So we're pretty happy with the collaboration.
I'm Laura Kilpatrick. I'm a postdoctoral research fellow in the lab of Professor Steve Hill at the University of Nottingham. So I'm currently using the NanoBRET technique developed by Promega to quantify the molecular pharmacology of the vascular endothelial growth factor receptor 2, or VEGFR2. And this is a receptor tyrosine kinase which is expressed on the surface of endothelial cells, and it's involved in blood vessel development and is also a very attractive anti-cancer therapeutic target. So the key benefit for using NanoBRET to investigate VEGFR2 pharmacology is that typically it's a receptor that's been quite hard to probe pharmacologically, as it's quite a large receptor, and the majority of your observations have been coming using just fragments of the receptor, not in its native membrane environment. However, the NanoBRET technique has allowed us to actually probe the receptor in living cells using full-length receptors for the first time. So we've been collaborating with the Advanced Technologies Group in Promega out in Madison in Wisconsin, and it's been a very fruitful and true collaboration. It's not just been a sharing of products from Promega's point of view, but also a sharing of common research ideas and critiquing the research as well as we've gone along. So one of the key advantages to this collaboration has been some of the research scientists at Promega have been able to develop for us a single-site fluorescently labelled version of one of the endogenous VEGF ligands, which has really allowed us to use this in conjunction with NanoBRET to specifically probe the pharmacology of VEGFR2. And another key advantage of this is we could use this single-site labelling technology to also probe our receptor tyrosine kinases in real time in living cells.
So I think the future of our research is to move more to native cells and at normal expression levels, which almost certainly will involve moving more into gene editing techniques. But the real advantage then is that we can apply those labelling strategies in drug discovery programs which are now using the right cell in the right environment, which of course we think will be much more disease relevant.